Welcome everyone to this cell signaling course. In previous classes, we have seen that how receptor-like kinases, a variant of receptor serine-threonine kinases, they are prevalent in the plant world and how they regulate every aspect of plant growth, development, differentiation, and any sort of regulation which it has to mediate between plant and its environment. And that is where we covered different signaling which we will see for plant kingdom. We will not be seeing in further classes any more reference to signaling in plants, because what we are going to discuss more in coming classes, apart from the receptor serine-threonine kinase signaling in animal world, we will see a receptor tyrosine kinase signaling which is absent in the plant world, as well as we will discuss a lot about G protein coupled receptor signaling, which is in very early age of its discovery and understanding in plant world. So we will in today's class see the signaling in animal world, and we'll start with the receptor serine-threonine kinase pathway for the continuity's sake. But if you recollect, what we have already dealt with is signaling in the living world, where we saw histidine kinase, whether it was canonical or the hybrid type. Here you are seeing the example of hybrid type, where this was an additional domain in your sensor kinase which needed the presence of another molecule which was in this class of histidine phosphotransperases, and then you had a response regulator which were basic or more complex or evolved. Basic one which had minimum receiver domain, and complex one had additional domain with respect to the response regulator's receiver domain. We saw examples of this aplenty in microbial world, whether it was bacteria or fungi we discussed. It is also prevalent in the plant kingdom, where we saw how different signaling downstream of cytokinin and ethylene was mediated by the receptor histidine kinase, which is actually part of two component system. What we saw in last couple of classes was receptor like kinases, which was serine-threonine kinase in plants, and how bulk of activity which plant carries out is mediated by receptor like kinase. If you recollect, we saw that how binding of ligand to its receptor activates the cytoplasmic domain, which has the specific kinase activity towards serine and threonine. This activated a soluble kinase which was in the family of RLCK. This activated RLCK was impinging on to the MAP kinase pathway, where we saw MAP3K getting activated, which in turn activates MAP2K, and this activated MAP2K in turn activates MAPK, which was sending signal to effector molecules. In previous class, if you recollect, we discussed about this transcription factor speechless, which was phosphorylated by the MAPK, and this was important for the epidermal cell transforming into the meristemoid cell. So in nutshell, we saw different kind of signaling in the plant kingdom. And if we talk about the receptor serine-threonine kinase, before that, if you think about serine-threonine kinase activity, this, if you recollect, I mentioned this is the most prevalent type of kinase activity in the living world. So let's see what all happens in case of receptor serine-threonine kinase in animal world. Before I enter into the details of the receptor serine-threonine kinase in animal world, let's draw some parallel which will help us build the case for receptor serine-threonine kinase in the animal kingdom. If you recollect, particularly for plants, we saw that extracellular domain of these receptor like kinases, one of the domain was leucine rich repeat domain which was present over there, and that was working through a set of molecule which in the previous class I told Werke 34, Werke 2, we saw the example of that, but similar family molecule, different Werkes, they serve as a transcription factor and they were responding to the pathogenic signal. If you remember the pathogenic signal, that was like chitin, peptidoglycan and flagellin. Exactly similar situation is present in the animal world, where LRR domain containing kinases or receptors which are called toll-like receptor, TLRs. There are multiple of them which are present in animal world and they are working through these serine-threonine kinase activity. Like this pathway was activating Werke, here this activation of toll-like receptor activates this molecule NF-kappa B. And if you recollect, this particular molecule was very important because it was regulated by its inhibitor I-kappa B. And this regulation is very important for activation of NF-kappa B, which is a strong transcription factor and regulates the, or actually generates peptides which are antimicrobial in nature, certain gases like NO and so on, so that the pathogenesis or the response to the pathogens could be mounted. So similar pathways starting from the receptor, if I say plants or animals, you have similar ligands which is chitin, peptidoglycan, flagellin. In case of animals you can have many more cues in addition to these, which here was working through Werke, and in animals it is working through NF-kappa B. So this particular discussion is about building the connect between the serine-threonine kinase activity which is seen in plant kingdom, where one of the receptor was mediating the process, to the animal kingdom, where we are going to see how receptor serine-threonine kinase works in animal kingdom. Does it follow exactly what we saw in case of plants, where we saw MAP3 kinase activity, the pathway getting activated, or there is something different or distinct in this case? So let's understand little bit more about receptor serine-threonine kinase. The name is evidently suggesting what exactly it will do. It is a receptor molecule which should be having a kinase activity towards serine and threonine residue, and this will be under the class of receptor protein kinase because this will phosphorylate the protein residue. And the serine-threonine kinase activity, this is present in a lot of soluble serine-threonine kinases, which makes it the most prevalent kinase in the living world, and you will see a lot of regulatory phosphorylation which are happening over there on these serine and threonine residue, which is important for activating the pathway, the signaling which one is understanding. If you think about receptor serine-threonine kinase, they are very important. Particularly many developmental processes are regulated by receptor serine-threonine kinase, and one of the key process is bone formation, and we will see how the discovery of initial serine-threonine kinase kind of receptor started, or serine-threonine kinase of activity started when people were studying the bone formation. This is key signaling pathway in immune system, where we saw example from NF-kappa B, which is generally inhibited by I-kappa B. Phosphorylation event ensures that this I-kappa B is degraded. This molecule moves to nucleus and binds to several sites. Depending on the signaling cue, it will ensure a particular transcriptional program is activated. And as I said, they are present in animal kingdom, but not to the extent we saw in case of plants. That is why in animal system the signaling through RSTK is not heavily contributing to diverse processes. So early discovery of this serine-threonine kinase receptor started when people were studying bone formation, and here it says that bone formation by autoinduction. So when people were working on this, the idea which came up was that new bone formation by autoinduction, which means that there are cells which signal to themselves and they work as a inductor cell, and that is how a new area where these cells will be present, they will give signal to make those cells as a bone cell. So there is something which is present in these inductor cell which communicates to the neighboring cell and induces a bone cell fate. Or if I can say, if you take extract from these cells and put in a new milieu where different types of cells are present, they will induce this pathway to make bone cells. And that is where some of these molecules were initially characterized as bone morphogenetic protein or BMPs. This included the soluble ligands as well as the receptor which participate in this bone formation ability. Early reports from different groups suggested that the bone morphogenetic proteins, they can be prepared by crushing the bone, preparing an extract from there and putting this extract to a new system where it will induce the bone formation. So there are some regulatory cues in the extract which is prepared from bone. And this can induce bone formation at a new location or sometimes referred as ectopic location. So like you saw in case of receptor serine-threonine kinase activity in plant kingdom, you will definitely need these components which are required for signaling. You will need the receptor, you will need the ligands, so this extract will definitely have those two, as well as you will need the effector molecules. I have been telling you that effector molecules play a very critical role in diverting the signaling in the desired direction. One cell type will have a given type of variant of effector molecule than what is seen in the other cell type. Now if you think about the receptor serine-threonine kinase activity and think about the receptor itself, what you recollect from the plant system was that they were often forming the heterodimeric assemblies, right? In one of the last class we saw that SCM was binding with BAC or SERK1 and BRI1, they were making the complex. So somehow in case of receptor serine-threonine kinase, heterodimeric structure formation is a preferred route. So does this happen in case of animal cells as well, or what if there is a different arrangement altogether? So let's see what is the situation in case of animal system. So here one step ahead, not just heterodimeric, but you have a tetrameric assembly of the receptor, which means that you will have receptors which are like this. In the membrane, you will have one set of receptor. If suppose this is one set of receptor, it is present in two copies. The other receptor probably could be of this type and present in two copies. Together they will make a functional receptor, and how many different receptor units are present here? 4 units. So these are heterodimeric type 1, type 2 coming together and making a functional tetrameric receptor. I must also emphasize again, which I have been telling, that receptor dimerization or oligomerization is one of the key event towards activation of the receptor. So the type 1 receptor which is there is phosphorylated. If I take this example here and I say this is type 2 receptor and this is type 1 receptor. So the phosphorylation happens in this type 1 receptor. And who will catalyze that? Type 2 receptor is catalyzing the phosphorylation of the type 1 receptor. So serine-threonine kinase activity is present in the type 2 receptor, which is ensuring that not only type 1 receptor is phosphorylated, even the type 2 receptor itself is phosphorylated, which we call autophosphorylation. What will happen downstream of these phosphorylation event? Remember that any signaling is dependent on the next messenger molecule, something which can amplify the idea that receptor is bound with the ligand. This has to be amplified, and for that the cell is dependent on the next set of messenger molecules, whether it is another kinase or adapter protein or something else. Here you would need specific adapter proteins to ensure that signal is amplified. In case of plants, you saw that some RLTKs, receptor like cytoplasmic kinases, they were coming and getting phosphorylated. Same thing will happen here. You will need an adapter molecule which will come in the vicinity and get phosphorylated. Now further what will happen? This phosphorylated adapter or next or the final effector molecule will oligomerize and be sent to the nucleus for signaling. When things go into the nucleus, largely we have seen that this ends up activating, inhibiting or regulating the transcription which is towards the response to the signaling which originated at the cell surface. So if we look at this, how this entire thing can be spanning out, that you have the membrane. If I can say that there is specific arrangement of the receptor in 2 copies. If I say this is your type 2 receptor, then you have another, this is your signal molecule which will be recognized by the type 1 receptor. So type 2 dimer, type 1 dimer, ligand binding is mediated by that. Now this type 2 receptor will phosphorylate here, and this will be taking the signaling forward. And if you think about different types of receptor, I started by saying that bone morphogenetic protein, this is through bone morphogenetic protein, through that we understood different components of the receptor serine-threonine kinase signaling. So the known receptors which are there in this family, and if we can say, we can broadly classify them. So there is one class which is called bone morphogenetic protein receptor. Then there is another molecule which is called activin type. We will see how this receptor plays very important role in mediating various aspects of immune signaling. Then there is an example of TGF beta. If you recollect, I have mentioned this earlier also. Tumor growth factor receptor. This is another type of receptor which is known to be present in the class of receptor serine-threonine kinase, and one very interesting receptor which is not that very well understood is called anti-Mullerian hormone receptor, or if I say AMH receptor. So remember, you have this, the names which I have given you, these are directly referring to the type 2 receptor. And as we have understood that receptor serine-threonine kinase is a heterotetrameric assembly of type 2 dimer and type 1 dimer. So with all these receptors which I am mentioning here, you will need type 1 receptor also, and many of them are part of this activin-like kinase. This will be in different isoform format. It will be making sure that type 1 receptor is available. It all sometimes depends on how these molecules have been identified, in what context they have been identified, and accordingly they have been named. So different type 1 receptor which will be making combination with BMP or activin receptor or TGF beta receptor or AMH receptor, they are broadly classified as ALK, activin-like kinase receptor, and then they can further be classified like, okay, what exactly is their combination in which they are present and in which they are signaling. Now, if you think about the next molecule which will be working over there, how the signaling is moving forward. So let's make a simpler diagram here, that where you have the receptor, and I say type 2 and type 1 receptor, binding of the ligand, we saw it will start the phosphorylation event, 2 will activate 1, phosphorylation happens. This phosphorylation brings first set of effector molecules in this sequence, and these are called SMADs. SMAD, this will undergo phosphorylation event. This phosphorylated SMAD will combine with another SMAD. Sometimes these SMAD which come directly in contact with receptor, they are called R-SMAD, meaning SMAD which connect with the receptor. This phosphorylated R-SMAD forms a dimer, and it also needs another adapter molecule which is SMAD4. Thus it will make a oligomeric assembly. This oligomeric assembly will now be targeted for translocation into the nucleus. And this nuclear translocation is something which will be much awaited event downstream of this receptor serine-threonine kinase signaling, where you have a group of molecule which are transcribed. This complex comes and bind to this promoter region, and it works through different proteins. Some of them are called P300 and CBP, through which they activate the signaling and ensure that end product of this region is, this particular activity is met. Once the signaling is complete, the molecule goes out. For that you will need dephosphorylation of SMAD event. R-SMAD must undergo dephosphorylation and thus moves out. And here it will be how the signaling, the entire signaling will be regulated in that case. What will be happening, that now consider a scenario where we are looking at nucleus here, and the receptor, the R-SMAD has come out of the nucleus, and this is dephosphorylated. This can be actually degraded by ubiquitination. as well as now you are looking other way around. So, this is your receptor and this is your signal. There are SMAD which actually compete with binding here and inhibit the R-SMAD binding, so that it cannot be phosphorylated and it cannot move inside the nucleus along with SMAD4. There will be enzymes which will be ensuring that any R-SMAD which is there, that is degraded also, something which you saw as a degradation here. Something similar can happen at the receptor end as well as there are molecules in the SMAD family only, like SMAD6 and 7. They will ensure that they compete and R-SMAD does not bind to that, or they will come along with the enzyme which will degrade the R-SMAD. This is how the signal is regulated. Now one question is there, why generally you will need R-SMAD to couple with SMAD4 so that it can go to the nucleus? And this is where the answer lies in the domain organization of these R-SMAD. So some of these SMAD will have nuclear localization signal. So SMAD4 you would expect to have a nuclear localization signal, or how things will come out of the nucleus. You will need nuclear export signal to be present. So R-SMAD also has the NLS, the SMAD4 also has the NLS. Together when they come in a complex, they make a much stronger entity which directs them inside the nucleus, and eventually with the help of another signal which is called NES, nuclear export signal. Once the job is done inside the nucleus and dephosphorylation event is preferred, you will have this nuclear export signal exposed, and this will take the entire complex R-SMAD and SMAD4 out of the nucleus into the cytosol where they will be degraded. So basically what you will notice is that the pathway which is being followed downstream of the receptor serine-threonine kinase in animals seems very different from what we saw in the plant kingdom. The kinase activity is same, but the pathway is following very different route. It all depends on what kind of molecules are downstream of the receptor to take the signaling forward, the signaling molecule, the messengers or the effector molecules. So what we have learnt in this class today is that receptor serine-threonine kinase, the signaling is prevalent in animals also. Lot of developmental aspects are regulated by them, but they use a very different activation mechanism which is different from MAP kinase pathway. Also here you have different types of receptor which come into a heterotetrameric form, 2 copies of type 1 and 2 copies of type 2. Together they make heterotetrameric assembly, which is the active form. And out of the 2 dimer, type 2 receptors are the ones which have the catalytic activity and activate the entire complex by phosphorylating type 1 receptor. So receptor serine-threonine kinase in animal world present a very different case than the plant world. Thank you.